Bismillahirrahmanirrahim. Assalamu alaikum. So continuing with the topic Neoplasia, today we are going to have the second lecture. So let's discuss some of the other topics. So the learning objectives of today's lecture are, at the end of this lecture, you will be able to trace the pathways for hematological spread of a metastatic cancer cell. You'll be able to use the concepts of growth fraction and doubling time to explain the growth of the cancerous tissue. You'll be able to describe various types of cancer-associated genes and cancer-associated cellular and molecular pathways. And you will be able to know about genetic events and epigenetic factors that are important in tumorigenesis. And finally, you will have some knowledge about cancer stem cells, angiogenesis, and the microenvironment in cancer growth and metastasis. So coming up to the first topic, which is invasion and metastasis. As you all know that this is a property of cancers. Unlike benign tumors, which are more localized and they are also surrounded by a capsule, cancers have a property in which they can invade and they can extend into the surrounding tissues. So this occurs by direct invasion and extension, number one. Number two, they can have seeding of cancer cells into body cavities, in which the cancer cells are shed into some of the body cavities like the peritoneal cavity, the pleural cavity, the pericardial cavity, and the joint spaces may also be involved. And these cancer seeding can also be associated with fluid accumulation in these cavities. For example, ascites, which is a fluid accumulation in peritoneal cavity, while pleural effusion is a fluid accumulation in pleural cavity. And lastly, metastatic spread can happen through the blood or the lymph pathways. Alright, so what metastasis is? Metastasis means it is used to describe the development of a secondary tumor in a location distant from the primary tumor. So it is actually the same tumor which is occurring in the primary tumor, but it is distant from the primary tumor. Let's say if the primary tumor is in ovaries, now this secondary tumor which is spread either via the blood or through lymphatic spread, it's going to travel through the lymphatic vessels or the blood vessels and reach a location where they're going to start invading and they're going to make a secondary tumor, and that's called metastasis. So now lymphatic spread and hematogenic spread. Lymphatic spread occurs via lymph vessels, while hematogenic spread occurs via blood vessels or blood. So when metastasis occurs by the lymphatic route, the tumor cells actually first lodge in the initial lymph node that receives drainage from the tumor site. So the first lymph node which receives the drainage from the tumor site is termed as sentinel lymph node, and the tumor cells first lodge in this node, and once in this lymph node the cells may either die. These are the what do you say, after coming into the lymph node, the cells may either die because of the lack of a proper environment, or they will grow into a discernible mass, secondly, or they may remain dormant for unknown reasons. And if they are able to survive, they will grow, and that cancer cells may spread from more distant lymph nodes to either the thoracic duct, and via thoracic duct they'll ultimately gain access to the vasculature. As you all know that through lymphatic vessels it can be spread to either lymph nodes, to thoracic duct, and ultimately to venous circulation. And hematogenic spread is via blood. So the cancer cells may enter the venous flow that drains the site of the primary neoplasm, and cancer cells may also enter tumor-associated blood vessels that either infiltrate the tumor or are found at the periphery of the tumor. And before entering the general circulation, venous blood from the GI tract, most of the organs from the GI tract, the pancreas, the spleen, they cross the liver via the portal vein and then they move into the heart. So the liver is therefore a common site for metastatic spread of the cancers that originate in these organs, because all of these organs are draining their venous blood into the liver via the portal vein, and from the liver it's then carried out to the heart. Okay, so now we're going to discuss the mechanism of tumors metastasis. So what happens is, for tumor metastasis, first of all, the most important thing is that cancer cell who has the ability to metastasize, as it has all the genes or all the mutations and all the properties that are important. After that, it should be able to break its connection from the primary tumor. And when it breaks the connection from the primary tumor, what is it going to do? Let's say these are the cancer cells, as you can see, and this is the basement membrane. This is the primary site of the cancer. And what happens is, a cancer cell becomes capable of invasion, okay, and then it expresses some surface adhesion molecules. Now this is a cancer cell which has become capable of invasion. So now it's going to express some surface adhesion molecules. As you can see here, these are the surface adhesion molecules. So these cancer cells are expressing these molecules, and these molecules are going to bind to the underlying extracellular matrix. Now these are binding to the basement membrane. And after adhesion, what are they going to do? Now these cancer cells also have some property of releasing proteolytic enzymes. And when these proteolytic enzymes are released, they're going to break down this membrane, and hence these cancer cells are then going to invade or pass through this disrupted basement membrane, and then they're going to enter into the blood vessel or the lymphatics. Here, this is the blood vessel, or this is the lymphatic. So they may enter the blood vessel or the lymphatic, and after gaining access to the blood vessels or lymphatic, they are going to survive its passage in the bloodstream, and after that they are going to emerge at a favorable location and then invade the surrounding tissue. Then they'll begin to grow and establish a blood supply, which is called angiogenesis. Angiogenesis means the establishment of new blood vessels and specific growth factors that promote proliferation of the tumor cells. So this is the mechanism which is involved in tumor metastasis. Now we are going to talk about tumor growth. So the rate of tissue growth in normal and cancerous tissue actually depends on three factors. Number one is the number of cells that are actively dividing or moving through the cell cycle. The second one is the duration of the cell cycle, and the last one is the number of cells that are being lost relative to the number of new cells being produced. So discussing about the first point, the number of cells that are actively dividing or moving through the cell cycle. So we need to know about two terms, the growth fraction and the doubling time. What is the growth fraction? It is the ratio of the dividing cells to resting cells in a tissue mass. And what is the doubling time? It is the length of time which the cancer cells take for the total mass of cells in a tumor to double. Alright? And so, as you know, that as the growth fraction increases, now growth fraction means if the growth fraction increases, that means that there are more number of dividing cells than the resting cells. I mean, there are more dividing cells. Then the doubling time will decrease. Ultimately, because of course, if there are more dividing cells, they'll take less time to create a mass of cells. So hence the doubling time will decrease. And when normal tissues reach their adult size, an equilibrium between cell birth and cell death is reached. But this does not happen in cancer cells. So what happens in cancer cells? Cancer cells, however, continue to divide until limitations in blood supply and nutrients inhibit their growth. So this ability for them to divide continuously is one factor in which they're growing in such large numbers. Okay, now discussing about the number of cells that are being lost relative to the number of cells that are being produced. So again, cancer cells do not die on the schedule, and growth factors prevent cells from exiting the cell cycle and entering the G0 phase. So this is again one property of the cancer cells which differentiate them from the normal cells, that they do not die and they do not exit from the cell cycle and do not enter the G0 phase. Okay, now talking about the genetic and molecular basis of the cancers. So as you all know that cancers should have some cause, and what is the most important pathogenesis of all these cancers? So normally speaking, we know that there are three pathogenesis. First one is genetic damage or mutations in the genes. As we all know that we say that cancers are caused by genetic damage or mutations, which with resultant changes in cell physiology that transform normally functioning cell into a cancer cell. Then second is epigenetic factors that involve silencing of a gene or genes. So some epigenetic factors that are going to silence, or they're going to, you can say that they're going to stop a gene or a number of genes. And cancer stem cells. There are some cancer stem cells as well. Okay, so considering the genetic properties, first of all, we're going to talk about some cancer associated genes. So actually, the cancer associated genes can be divided into two categories: genes that have an overactivity and genes that have an underactivity. So genes that have overactivity are called proto-oncogenes. All right, and these are actually normal genes that become cancer-causing oncogenes if mutations happen in these genes. So proto-oncogenes encode for normal cell proteins such as growth factors, growth factor receptors, etc. And an example of proto-oncogene is CMYC is linked with oral squamous cell carcinoma. So actually when a gene has an overactivity, it is supposed to be called a proto-oncogene, and this causes cancer. While there are some genes that have underactivity, and due to underactivity of those genes, they're going to cause cancers. For example, we have tumor suppressor genes, which by being less active create an environment in which cancer is promoted. All right, so tumor suppressor genes actually are retinoblastoma genes. There are two genes of tumor suppressor genes: retinoblastoma gene and TP53 gene. So now talking about retinoblastoma gene, RB gene, which normally prevents cell division. So as this is a tumor suppressor gene, that means it's going to prevent cell division and it's going to suppress the tumors. But if there is underactivity or loss of this RB activity, it may accelerate the cell cycle and lead to increased cell proliferation, and hence it may cause cancer. While the other gene, the TP53 gene, which normally becomes activated in DNA damaged cells to initiate apoptosis. What it does is, normally when a cell is damaged, when a cell is damaged and the DNA is defective, it kills the cell by apoptosis so that it does not cause any harm to the body. But when there is underactivity of this gene, what happens is it may increase the survival of these DNA damaged cells, and these DNA damaged cells will further proliferate and form cancer. All right, okay, so the next topic is that the genetic events that leads to oncogene formation or activation. So I told you that there are two gene activities, the gene overactivity and gene underactivity. So we are going to talk about the events that are going to lead to oncogene formation or activation, this thing here. Okay, so first of all, we'll talk about point mutation. Point mutation is actually a process in which there is a single nucleotide base change due to an insertion. Insertion means either there is a single nucleotide added into the previously occurring thing, or it is deleted, or it is substituted with another base. So this is a point mutation in which there is a single nucleotide base change, which can be either due to insertion, deletion, or substitution. An example of an oncogene caused by point mutation is the RAS oncogene, which has been found in many cancers. Now this is one event. Second event that can lead to gene activation, oncogene formation or activation, is gene amplification. Here, gene amplification. So multiple copies of certain genes may lead to overexpression with higher than normal levels of proteins that increase cell proliferation. For example, the human epidermal growth factor receptor 2, which is called HER2/neu gene, is amplified in many breast cancers. And thirdly, chromosomal translocations. Now, to discuss about chromosomal translocations, you have to look at this picture. Now, we are going to talk about the two examples, the A and the C. So this is for the chronic myeloid leukemia, CML, which is the cancer of the blood, and this is of Burkitt lymphoma. All right. So what happens is, in chromosomal translocations, now you can see two chromosomes here, the chromosome 9 and the chromosome 22. So translocation means the transfer of a part of chromosome from one chromosome to other chromosome. So in case of CML or chronic myeloid leukemia, what happens is, on the long arm of chromosome 9 and on the long arm of chromosome 22, there happens to be a break, and a part of this chromosome is translocated into the chromosome 22. And a new chromosome is formed, which is called Philadelphia chromosome, and which has a fusion gene, which actually encodes this protein, BCR or ABL protein, which helps in the pathogenesis of the CML cancer. All right, so this is an example of chromosomal translocation. And another example of chromosomal translocation is here in Burkitt's lymphoma. There is a chromosomal translocation of between chromosome 8 and 14. And what happens is, and chromosomal breaks involve the long arms of chromosome 8 and that of chromosome 14. And this CMYC gene on the chromosome 8 is translocated to a region on chromosome 14 adjacent to the gene coding for the constant region of an immunoglobulin heavy chain. And hence this new chromosome is formed, which has this oncogene, MYC oncogene, and hence this results in pathogenesis of the Burkitt's lymphoma. Okay, now genetic events that lead to loss of tumor suppressor gene function. Okay, the other thing that we discussed here was gene underactivity. So what are the events that are involved in that underactivity? So actually, as you know, in this case, we only had one mutation. It can be either a point mutation, a gene amplification, or chromosomal translocation. But for tumor suppressor gene function loss, we need a two-hit hypothesis. Two-hit hypothesis means there should be two mutations of carcinogenesis. So the malfunction of tumor suppressor genes may require two hits to contribute to total loss of the function. The first hit may be a point mutation in an allele of a particular chromosome. Later, a second hit occurs that involves the companion allele of the gene. In hereditary cases, the cases in which the patient acquires a mutation from the parents, it's called hereditary cases. The first hit is inherited from an affected parent, and so is therefore present in all somatic cells of the body. While in sporadic cases in which the mutation is not inherited, that means it's not taken from the parents. Both mutations or both hits occur in a single somatic cell, and whose progeny then forms the cancer. So you can look at this picture here. This shows that how that two-hit hypothesis works. So this in A you can see that a child has retinoblastoma, and here again a child has retinoblastoma. But in this case, the child had inherited retinoblastoma. That means his parents were involved. So he actually had a mutation, inherited mutation in his genes. So this was the first hit, and then the somatic mutation, which was the second hit, he acquired the second hit, and after two hit mutations, he developed retinoblastoma. But in case of sporadic what happened is he did not acquire any from his parents because there was no history in his parents. So they were normal genes. What happened? There happened to be a somatic mutation, which was the first hit, which turned into a somatic mutation here. Then the second hit happened, the second mutation happened, and again he acquired 2 hits or 2 mutations and he developed retinoblastoma. Okay, so now we're going to talk about epigenetic mechanisms. So in addition to mechanisms that involve DNA and chromosomal structural changes, there are molecular and cellular mechanisms termed as epigenetic mechanisms, which involve changes in the patterns of gene expression without a change in the DNA. Epigenetic mechanisms, as I told you earlier, may silence genes such as tumor suppressor genes, so that even though the gene is present, it is not expressed and a cancer suppressing protein is not made. So now example of epigenetic silencing is methylation of the promoter region of the gene, a change that prevents transcription and causes gene activity. Okay, so the next topic is molecular and cellular pathways. So we'll talk about some of the mechanisms or some of the pathways that actually aid in causing cancers. So among them, the first one is DNA repair defect. So this is a normal phenomena that occurs, the DNA repair phenomena. For example, if a cell is exposed to any toxic agent, what happens is it causes DNA damage in the normal cell, and there is a normal mechanism in most of the cells in which the DNA repair genes act and they repair the DNA. But if there are mutations or any other thing that results in the DNA repair genes defect, then what happens is this DNA damage results in failure, and hence it results in activation of growth promoting oncogenes and inactivation of the tumor suppressor genes, and thirdly alterations in genes that control apoptosis, and this will ultimately lead to unregulated cell differentiation and growth that will cause malignant neoplasm. So this is actually a pathway that shows that how DNA repair defect will affect and will cause a neoplasm. Okay, the second pathway is defects in growth factor signaling pathways. So this shows a normal pathway for gene regulating cell growth and replication. As you can see here, this is a growth factor in this picture, and this growth factor binds with a growth factor receptor here, the one shown in red, and it activates this receptor after the growth factor binds to its receptor. Now this activation of the growth factor receptor then further sends signals across the cell membrane into the cytosol, and these signals are sent via signal transducing proteins, and when they come across to the nucleus, it hence forms the transcription, or it also helps in the progression through the cell cycle. So this is a normal process again, and these signal transducing proteins that transmit the growth promoting signal to the nucleus are actually a number of proteins, and they exert their effects with the help of these enzymes called kinases. These kinase enzymes actually phosphorylate proteins. As you can see that these phosphorus are added to the proteins, and this way they are going to activate them and send the signals to the nucleus. So if this pathway has a defect, or if there is a defect in this protein or maybe the whole pathway, it will also result in oncogenesis or tumor formation. Now the third is evasion of apoptosis. Apoptosis is considered a normal cellular response to DNA damage, and loss of normal apoptotic pathways may contribute to cancer by enabling DNA damaged cells to survive. Now example of this is that high levels of the anti-apoptotic proteins BCL2 that occur secondary to a chromosomal translocation in certain B-cell lymphomas. Now, again, in this case, you'll know that apoptosis is important to repair the damaged cell and or to engulf the damaged cell. And if this normal mechanism is again defective, then of course it will lead to cancer. And again, if there is a defect in cellular senescence, senescence means aging. Normally what happens is normal cells, they if they have any defect or any damage, they result in aging, and hence they are exited from the normal process. But cancer cells don't have this property, and cancer cells are immortal as they have high levels of telomerase that prevents cellular aging and senescence. So again, there's a defect in cellular senescence that leads to cancer formation. Now the fifth one is development of sustained angiogenesis. As I told earlier, angiogenesis means the formation of new blood vessels, and this promotes this actually promotes cancer formation and cell proliferation. So increased production of angiogenic factors or loss of angiogenic inhibitors. Now this is important. The normal TP53 gene seems to inhibit angiogenesis by inducing the synthesis of an anti-angiogenic molecule which is called thrombospondin 1. So now this is a normal process, but with mutational inactivation of both TP53 alleles, if there is defect in this gene or if there is a mutation in this gene, what happens is, because these mutations occur in cancers, and then what happens, of course, the levels of this molecule, anti-angiogenic molecule, which was called thrombospondin 1, drops, and hence it will tilt the balance in favor of angiogenic factors. That means more angiogenesis will occur, and it will help the cells to proliferate more, and hence cancer formation. Lastly, we have invasion and metastasis. The MET proto-oncogene, which is expressed in both stem cells and cancer cells, is a key regulator of invasive growth. Now we're going to talk about the role of the microenvironment. Actually, the microenvironment of the cancer cell consists of multiple cell types, which includes macrophages, fibroblasts, endothelial cells, a variety of immune and inflammatory cells, the extracellular matrix, and the primary signaling substances such as cytokines, chemokines, and hormones. Alright, and this microenvironment is also important for the tumor growth and metastasis as well, and angiogenesis, and of course for metastatic tumor survival as well. Now, the example of this microenvironment is signaling of the cytokine transforming growth factor beta, TGF beta, is known to be important in the cellular pathway leading to cancer cell formation. Okay, lastly, we're going to talk about carcinogenesis. Now, carcinogenesis is actually, we're going to talk about the steps that are involved in cancer formation. The steps that are involved to transform a normal cell into a cancer cell, that's called carcinogenesis. And there are actually 3 steps: the initiation, the promotion, and the progression. So the initiation is the first step. As you can see here, that this is a normal cell. What happens is, when there is a carcinogenic agent or there is a stimulus, which can be a chemical, it can be radius, or it can be virus. Due to this carcinogenic agent, when the cell is exposed to this agent, there are some changes that occur in that particular cell. So this is the first phase, which is called initiation, and that changes include DNA damage and cell mutation. After that, that cell starts replicating, and the cell proliferation starts. As you can see here, that this mutated cell starts growing, and that when that stage, when these cells start growing, it is called the second stage, which is the promotion. Promotion is the second step that allows for prolific growth of the cells, triggered by multiple growth factors and chemicals. So this is the second stage, allowing the growth of the cells, and 2 important things are to be mentioned in this stage, that is the growth factors and the chemicals, which help the promotion of the cells. Lastly, we have the progression stage. in which the cancer cells have the ability to invade and to metastasize. So this is the last step of the process that manifests when tumor cells acquire the malignant phenotypic changes that promote invasiveness, metastatic competence, autonomous growth tendencies, and increased karyotypic instability. So that was all about the second lecture on neoplasia.